Here are Shade 8 high-yield USMLE-style questions, focused on the cell cycle, cyclins, cyclin-dependent kinases, CDKs, tumor suppressors, and cell types. Question 1. Which of the following is a key function of cyclin-dependent kinases, CDKs, in the regulation of the cell cycle? A. They are activated by phosphorylation to control cell cycle progression. B. They are constitutively expressed and active during the cell cycle. C. They require binding to specific cyclins to become active and regulate cell cycle transitions. D. They inhibit the phosphorylation of tumor suppressor proteins like RB. E. They are inactivated when bound to cyclins. Answer. They require binding to specific cyclins to become active and regulate cell cycle transitions. Explanation. CDKs are constitutively expressed but are inactive when not bound to cyclins. Once cyclins bind to CDKs, they become active and can phosphorylate target proteins to regulate transitions between cell cycle phases, including G1S and G2M checkpoints. Question 2. A mutation in the RB protein results in its hyperphosphorylation, leading to unrestrained cell division. Which phase transition of the cell cycle is most affected by this mutation? A. G0 to G1, B. G1 to S, C. S to G2, G2 to M, EM to G1. Answer. B. G1 to S. Explanation. The RB protein acts as a tumor suppressor and regulates the G1 to S phase transition by inhibiting the E2F transcription factor. When RB is hyperphosphorylated, it is inactivated, leading to uncontrolled progression from G1 to ester, which can contribute to unrestrained cell division, as seen in cancers and conditions like Li-Fraumeni syndrome. Question 3. Which of the following tumor suppressors inhibits CDK activity by inducing P21 expression in response to DNA damage? A. P53, B. RB, C. EGF, D. Cyclin D, E. PDGF. Explanation. P53 is a tumor suppressor that plays a crucial role in the G1 checkpoint. Upon DNA damage, P53 activates the expression of P21, which inhibits CDKs, thereby halting the cell cycle in G1 to allow time for DNA repair. If DNA damage is irreparable, P53 can initiate apoptosis. Question 4. Which of the following cell types is permanent and typically remains in G0, regenerating only from stem cells? A. Hepatocytes, B. Lymphocytes, C. Neurons, D. Bone marrow cells, E. Skin cells. Explanation. Permanent cells like neurons, skeletal muscle, cardiac muscle, and RBCs typically remain in G0 and do not proliferate. These cells regenerate only from stem cells or undergo limited repair when damaged. Question 5. A mutation in a cyclin-dependent kinase, CDK, results in the cell bypassing the G1 checkpoint and proceeding into S phase without proper DNA damage repair. Which of the following tumor suppressors is most likely affected by this mutation? ARB, B. P53, D. Cyclin B, E. EGF. Answer. B. P53. Explanation. P53 is critical for monitoring DNA integrity during the G1 checkpoint. In response to DNA damage, P53 halts the cell cycle by inducing P21, which inhibits CDKs. If P53 function is impaired due to a mutation or inhibition, the cell may bypass the checkpoint and progress into S phase despite DNA damage, increasing the risk of genomic instability and cancer. Question 6. Which of the following growth factors plays a key role in the transition of cells from G1 to S phase? A. Insulin, B. PDGF, C. EPO, D. EGF, all of the above. Answer. All of the above. Explanation. Growth factors like insulin, platelet-derived growth factor, PDGF, erythropoietin, EPO, and epidermal growth factor, EGF, bind to tyrosine kinase receptors and promote the transition from G1 to S phase. These growth factors stimulate cellular division and are essential for tissue regeneration and repair. Question 7. During prophase of mitosis, which of the following occurs as part of the M phase? A. DNA is replicated. B. The nuclear membrane reforms. C. Chromosomes align at the metaphase plate. D. Chromatin condenses into visible chromosomes. E. The cell undergoes cytokinesis. Answer. Chromatin condenses into visible chromosomes. Explanation. During prophase, the chromatin condenses into distinct chromosomes that can be seen under a microscope. The nuclear membrane begins to break down in preparation for metaphase when the chromosomes align at the metaphase plate C. Question 8. Which of the following cell types is most affected by chemotherapy as they have the shortest G1 phase and divide rapidly? A. Hepatocytes. B. Neurons. C. Skin cells. D. Bone marrow cells. E. Lymphocytes. C. Skin cells. Explanation: label cells, such as skin cells, gut epithelium, and bone marrow cells, are actively dividing with a short G1 phase and are most affected by chemotherapy, which targets rapidly dividing cells. These cells constantly cycle through the cell cycle, making them vulnerable to the cytotoxic effects of chemotherapy. These questions